Now we are going to start a new chapter. Chapter name is Principle of Inheritance and Variation. So in this chapter we will study mainly the pattern of inheritance and variation. What is actually inheritance? Inheritance is actually the process of transmission of characters in the form of genetic materials from the parents to the offsprings through reproduction process. So the transmission of characters as DNA molecules or genes from parents to the offsprings through sexual process of reproduction or asexual process of reproduction is known as inheritance. And actually what is variation? Variation is just opposite the term of inheritance. Variation means the dissimilar characters or the characters which are present in the offsprings but which are absent in the parents. That means the differences of the characters appearing in the offsprings and absent in the parents. The character differences between parents and offspring is known as variation. So character similarity between parents and offspring known as inheritance, and the character difference between parents and offspring is known as variation. So now we will study the details of this process. Actually, in the principle of inheritance and variation is proposed by one famous scientist whose name is Gregor John Mendel. Gregor Mendel, who proposed the first initiation for discovering the pattern of inheritance and variation. So this Gregor Mendel is actually a priest, and he conducted some experiments on a plant, on a pea plant. Its scientific name is Pisum sativum. Pisum sativum plant is a pea plant. He selected this species of pea plant with the different varieties or different strains. Actually this pea plant is having different varieties. So he selected different types of pea plants based on character difference. So what are the characters he noticed here for describing different varieties? That we will study. Mainly he noticed the character of this pea plant. It is a pea plant. In this pea plant, height was a character. Then flower color was a character. Then then seed color was another character. Then pod shape was another character. Then pod color was another character. Like that, he selected actually 7 characters total. So 7 characters he selected. Each characters are having 2 variations or 2 conditions, 2 opposite conditions. For example, height. Height is a character. Height may be in 2 conditions. Like if height is long one, that means tall. If height is very short, then that is dwarf. So if you are taking the character height, it itself has 2 forms. One is tall form, that is also a height. Height in the high level, that is tall, and height in the low level, that is dwarf. Again, if you are taking flower color may be white. In some Pisum sativum or some pea plants, the flower color was white. But in some other species, the same species but another variety, some other varieties of pea plants, they were having the flower color red. So flower color is a character. It itself has 2 forms, 2 alternative forms, either white color or red color, both are color. So that is a common character, flower color, and 2 variations are. Likewise, height is a common character, 2 variations are there, tall height or dwarf height. Like that, each and every 7 characters having 2 variations. One is just opposite of other. Like that, 2 variations were there. So actually if you are going to such variations, then we can notice actually 14 variations, because totally general character number is 7. So alternative form of each and every characters, totally it will come in the 14 number. So that are the characters he selected. This Gregor Mendel selected for his experiments on the pea plant Pisum sativum. And based on, he selected some plants and he crossed. Suppose for example he selected pea plant, tall plant he selected, then another dwarf plant he selected, and its pollen grains. Pollen grains are male gametophyte. Already we studied in the angiosperm reproduction chapter. So pollen grains or male gametophyte selected or he collected from this particular plant, and he pasted that pollen grains on the stigma of this dwarf plant, and artificially he conducted the situations for fertilization, and he collected such seeds, and again he made the new generation, and he observed the character. What are the character similarity of that new generation or offsprings or young ones with this both of parents? What are similar characters? What are dissimilar characters? Whatever characters appeared as similarity, such characters are known as inheritance, and whatever characters are different compared to between the parents and offspring, that is known as variation. So based on that experiments only he concluded all these points, and based on the experiment on this Bison plant he made some law. That law also we will study in detail one by one later on. So now we are going to study some more terms related with his experiment. First one, the plants which are selected for crossing, they are known as parents, parent plants. And what plants which are produced through fertilization or crossing, that is known as offspring. Parents are producers and offspring is product. So that is a character. Then again, this offsprings, now here you can see this plant, this tall plant we can consider as the first. From this plant he collected pollen grain. Pollen grains are male gametophyte, so we can consider this is the male plant. And here he collected that pollen grains and fused with, he artificially made the conditions for fusing it with the egg of this plant. So egg is actually from this plant. So this is the mother, this is the male plant, and this is the female plant. So both male and female plants you can see, both we are calling it as parents, and the seed collected through crossing process, and the seeds are providing next generation which is known as offspring. So what all seeds are collected and made first generation. First generation is known as first filial progeny or F1. F1 means first generation or first filial progeny. F means filial. F means filial. Filial means offspring. Offsprings are different level. We can see here. Now one type of offsprings will be developed here, that is known as first filial. So it is F1. Again, if you are crossing the pollen grains of such plants with the same egg, that means the same flower egg. That means self-pollination. We already studied self-pollination and self-fertilization. If you are conducting experiments like this, then again we will get seeds, and if you are sowing that seed, again we can produce next generation, that is known as F2 generation. Again, if you are collecting seeds and making next generation, then third generation will generate, and that is known as F3. F means filial. Filial means generation. F1 means first generation. F2 means the second generation, like that it is going. So we can see filial, or we will represent the generation through the F1, F2 symbol code. So now we are going to see the actual experimental situation or experimental process of Grigor. What was the experiment and how he has done that experiment. Mendel's first experiment is known as monohybrid cross. Mono means one, hybrid means it is a form of gene, hybrid gene or new type gene combination. New type gene combination is known as hybrid combination. So we will see that hybrid formation in this crossing time. So that is why it is known as monohybrid cross. Monohybrid cross, in short meaning, character of only one character is assessed in this crossing process. So one character only assessed. So it is also known as inheritance of one gene. Inheritance of one gene. Suppose any character, one character only assessed here. Already we studied that Mendel selected 7 characters with 2 alternative form of each character for his experimental purpose. So out of that 7 characters, any one character we can select here. Suppose height. If you are selecting height, then based on height, 2 alternative forms are there. So one is tall plant and another one is dwarf plant. So what he did is actually he selected one, this Gregor Mendel, who selected one tall plant, and he also selected one dwarf plant, and one plant he considered as a male plant and another one is considered as a female plant, and these both parents were true breeding parents or true breeding plants. True breeding, or it is also known as pure line plants. What is the meaning of true breeding or pure line? That means if you are making self-pollination of this particular plant, then generation after generation all plants will be in the tall level condition only. Suppose if we are here doing the self-pollination process generation after generation, then all plants are in the dwarf conditions only. That means in between dwarf condition there is no tall plant formation, or in between the tall plant formation of here there is no formation of dwarf plant. That means same type of plants are only produced through self pollination and self-fertilization process. Such a condition is known as pure plant. For example, the tall plant, this tall plant is taking, suppose this tall plant, and here flowers are here. Now if we are taking one artificial container and covering this plant, so that outside pollen grains are not able to come to this flower. So there is no possibility of cross pollination. So here then what will happen? Here bisexual flowers are here. In this bisexual flowers, anthers are there. From anther, pollen grains will be released. That pollen grain will come to this stigma, and fertilization will pollination will take place, and afterwards that male gamete will be released to the female ovary, and their fertilization will take place. So here actually fertilization of gametes which are produced in this plant itself or in this flower itself, such a type fertilization is known as self-fertilization. So here there is no chance of cross fertilization. That means it is enclosed by a bag. So outer pollen grains or outer other agents or other factors cannot able to come to this flower. So there is only one chance, that is self-pollination or self-fertilization. And after that self-fertilization, seeds are produced in this flower, and if you are collecting such seeds and making next generation, then that plant will also tall. Again, if you are covering that plant with artificial condition and making and selecting its seeds and putting it in the soil and then pouring water, then what will happen? Third generation will also tall. That means generation after generation there is no character change in the case of height in these plants. So we can say these plants are pure breeding or true breeding or pure life. There is no impurity of character in that case if we are conducting self-pollination only or self-fertilization only. So such characters are known as true breed. He selected true breeding character parents for his experimental purpose, and there we can say this tallness is a character, dwarfness is also a character. Both characters come under the category of height, and here tallness and here dwarfness, both are characters of height appeared in the body, and it is controlled by gene. So one character is controlled by a gene. Once again, one character is controlled by one gene. So one character is controlled by one gene, and one gene has two forms. That two forms are known as alternative forms or alleles. One gene has two alleles. This one will be one allele and this another will be one allele. So two alleles together known as one gene. That is a character. So here in each and every diploid condition, two alleles of a gene is present. Two alleles present in one diploid condition. In haploid condition, only one allele is present. So here we can represent in this is tallness, this is a plant, this is a diploid condition plant. So here also height has one gene and one gene has two alleles. Alleles are part of gene. Two parts are there for one gene. They are known as alleles. Alleles number is 2 in most of the genes. Sometime in some genes number of allele may be high. That is known as multiple allelism or multiple allele condition. That is in some cases only or some characters only that condition is existing. That we will study later, but now we have to concentrate as the two alleles and one gene concept. One gene has two alternative form and they are known as alleles. Here tallness is a character. That gene tallness is a physical character. We can see that character whether it is tall or dwarf. So whatever characters we are observing through our eye, such characters are known as physical characters or phenotype. Pheno means physical. So what is phenotype? Phenotype means physical characters observable through our naked eye. Such characters are known as phenotype. It is actually controlled by genes. Genes are made by DNA in the chromosome that is located in the nucleus of the cell, that is invisible. By naked eye we can't see any gene, because if you are looking into one plant we can see its leaves, stem, roots, etc. only. There is no gene there, but actually that stem, leaf, root, everything made up by cells. In the center of one cell a nucleus is present. Inside the nucleus chromosomes are present. In the chromosomes DNA are present. DNA is the actual made up molecule of gene. So genes are there but it is invisible to our naked eye. So with the help of electron microscope only we can see such genes or DNA molecules. So actually DNA and genes are there, that is controlling or making these physical characters. So physical characters only observable, but genes are there. The genetic condition or genetic character or genetic constitution is known as genotype. Geno means genetic or gene. Type means constitution. So genetic constitution of one organism is known as genotype, and physical constitution or physical characters of one organism is known as phenotype. So likewise, this height is a physical character. We can see whether it is tall or dwarf plant. So it is a physical character or phenotype. So tallness is a physical character. So if you are writing it as tall, then that is phenotype. And actually it is controlled by some genes. Genes are not visible, but we can represent by using different letters. Here tallness is a physical dominant character. So we can represent that gene by the letter T. And already we studied one character is controlled by one gene. One gene has two parts. That parts are known as alleles. So we have to represent minimum two letters, because two parts are there or two letters are there for one gene. So here this tallness is a true breeding character or pure line character. So we have to represent the same type of letter, capital T, again one more capital, because both alleles are same type. And that condition is known as pure line condition. And here we can see its genes are present. This dwarfness is also controlled by one gene. One gene has two forms, so two alleles. So minimum two letters we should use here also to represent this dwarf condition. But here dwarfness is a small character compared to tallness. Tallness is a big character and dwarfness is a small character. This small character is considered as a recessive character or little silent character. So inactive or less dominant character is known as a recessive character, and a recessive character compared to dominant character, recessive character is a small character. So it is represented by the respective small letter of the capital form or dominant form of gene. Here two capital letters are used. So here dwarfness is a small form of height, tallness is a big form of height, dwarfness is a small form of height. So we can represent the two small letters to represent the genetic constitution or genotype of this physical character dwarfness. So we can take two small letters, and then what will happen here is, now this capital T and capital T. This capital T is one allele and this capital T is another allele. These two alleles together make up one gene. This two allele condition is now known as one gene. This is a gene, both together known as one gene, and this is part of the gene, this is part of the same gene. Here also like that, both small t together known as a common gene, and first small t is a part of that common gene and second small t is a part of the same common. So that, then what will happen? We have to understand, this is the diploid condition. This is the diploid. This capital T is a gene or allele which is present in one chromosome. Already we studied it. Chromosomes are located in the nucleus. Chromosomes are located in the nucleus of the cell. And we have to come to one point. Two similar types of chromosomes are present in the diploid condition in our cells and in plants also. The diploid condition, two similar types of chromosomes are present. So here if you are taking this is considered as one chromosome. This type of one more chromosome is present in the same nucleus of the cell. So we can say these two chromosomes are having same physical appearance, structure, shape, etc., etc. That means similar chromosomes. They are almost similar in many characters or many aspects. They are similar, so they are known as homologous chromosomes. Homologous chromosomes. What is homologous? Homo means same or similar. Logos means positions or locus. Logos means position or locus. So this is from here to here one position, from here to here another position of gene, from here to here another position of gene, like that. These are various positions, various chromosome is a combination of many genes. So many genes are having many locations within the chromosome. Here two chromosomes are similar in many characters. So similar chromosomes are known as homologous chromosomes. And again in the same nucleus itself, one chromosome may present in this shape. If you are looking into this chromosome and this chromosome, by physical looking itself, definitely we can distinguish both of them are non-identical category or non-similar, dissimilar chromosomes. This is not similar to this chromosome because its shape, structure and appearance, everything is different. So this chromosome and these chromosomes are not similar type of chromosomes. Such dissimilar chromosomes are known as non-homologous chromosomes. Non-homologous chromosomes. So why we are explaining this concept here? Because in order to represent the location or position of these two alleles of a gene, this tallness is controlled by one gene. That gene has two parts. One is this capital T form and another one is this capital T form, and this capital T is located in this chromosome, and this another capital T is located not in the same chromosome but in the another member of that homologous chromosome. So that is present here, that we can represent here. So this is a diploid condition, 2n condition. So diploid condition means each and every chromosomes are having two members or full number of chromosomes are present. So here two chromosomes are present. This one is, both are known as homologous chromosome. This is the member of the homologous member, this is another member of this homologous. So here capital T is present here, here capital T is present in the opposite member. So both are same type of chromosomes, so they are known as homologous chromosomes. Here both alleles are present in the different members of the homologous pair. So what will happen when this cell will be producing gametes? Gametes, we know that this is diploid condition, and when this cell or this body will be producing haploid gamete. Gametes are haploid. That means only one member of this homologous pair will be coming to the gamete. So these two members will be separated. These two members will be separated in the division time of meiosis for gamete formation, and only one member will be coming to one gamete. Only one member will come to one gamete. So gamete is having only one member. So this may come with this capital, and this will go to another gamete, because through one meiosis 4 cells are produced. Through one meiosis 4 gametes are produced, or 4 haploid cells are produced. So each member of the homologous pair will be going to different gamete. Each gametes are n condition only. That means only one allele is present. One gene has two parts or two alleles, but two alleles are present in the diploid condition only. In the gamete, when they are producing gamete through meiosis process, only one allele will be coming to that particular gamete. So here we can represent two types of two gametes are formed. One gamete with this capital T. Gametes are represented by the symbol of a circle, because gametes are round structures. Okay. Then this particular allele will be going to another gamete. So both are same type of gametes produced here. So we need to represent only one gamete, because both are same type of gamete. If gametes are same type, then only one time we need to take it for representation in the picture or experimental process. Here also likewise, what will happen? This is the diploid condition, or in the plant body only this condition is existing. In the gamete only one allele will be coming. In the gamete only one allele will be coming. So we can that allele will in the gamete will be always small t condition. So consider this is a male gamete. This is a male gamete. This is a female gamete. That means egg. This is the egg and this is the male gamete. Then both are fused through fertilization process, and a zygote will be formed. In the zygote is this is n condition, this is also n condition. Both n will be combined to form 2n condition in the zygote cell. So in the zygote cell, what are the alleles will be coming? This capital T will be coming, and this small t will also come. So diploid condition again established here, and this is a plant. In this plant, two homologous chromosomes are present. In one homolog member, capital T is present. In the same in the another member of the same homologous chromosome, not capital T, but this small t is present. Again, when they will produce gamete, at that time both will be separated, and here two types gamete will be produced. Sometimes gamete with the capital T allele and gamete with the small t. So two types of gametes are produced, but in this plant only one type of gametes will be produced, because all gametes will be having small capital T allele, definitely, because there is no small t condition here. So all gametes will be carrying capital T allele, but only one in number, and this that condition is represented as n condition or haploid condition, and here only one small t will be coming to all gametes. So here this is male gamete, this is female gamete, fertilization took place, so diploid condition is established. What is the character of the gene? You can see here. First form, one gene has two alleles. This is the first allele, this is the second allele. What is the nature of first allele? First allele is capital T and second allele is small t. Capital T itself is meaning that is the dominant character. Dominant character means that character is much visible compared to its recessive character. Small t is the recessive, that means small t is representing dwarf character. Capital T is representing tall character. If both are combined or mixed in a common plant, then only one character will be expressed, and that will be whatever the allele is present as capital form, that character will appear in that plant. Here you can see capital T is present here, small t also present here. Because of the presence of capital T, this plant will be a tall plant. Suppose both alleles are small t like this, then plant will be definitely a dwarf. Both alleles are capital T form or capital form, then that plant will be tall. At least one allele also capital, another one is small, then also plant will be tall. Why? Because this is more expressive form even in the presence of small t. Small t is inactive one. It can't make its character in the presence of its counterpart of dominant form. Dominant form is capital form. Recessive form represented by small letter. Dominant form represented by the capital letter and recessive form represented by the small letter. Okay, so now we got the first seed or first generation. This first generation is now represented by symbol F1. Now we got F1. Now we are going to conduct a self fertilization process in the same plant itself, so that we will get F2. Let's see the condition of F2 here. In the F2 condition, what we are going to do, self fertilization will take place. That means here gamete fertilization is taking place by the fusion of two opposite gametes. Male gamete and female gamete should be there. What are the male gamete possibility to form here? What are the male gamete possibility? Male gamete with a capital T and male gamete with a small t. Again, if you are going to, these are male gametes both. Again, if you are going to female gamete, what are the conditions? Egg with a capital T and egg with small t condition. So these are the conditions. So we have to make this chart, or otherwise we have to make a table, then only we can see its level of crossing.